Cells copy their DNA in a process called DNA replication. So, before a cell divides, remember meiosis, cell division, its DNA first must be copied. So, I want you to think about a couple of questions. How do you think the double helix structure of DNA makes this possible for DNA to be copied? What do you think might happen if one of the nucleotides is damaged or chemically altered just before the copying process? And the last question I want you to think about is how might this damage affect the DNA that's inherited by each of the daughter cells after cell division? In this photo here, you see these 3D models of a skull that's being represented using math to be built again on the computer. So these scanners are scanning this 3D model and replicating the model on a computer. So if computers can do this, how is this possible in our DNA? So let's remember our structure of the DNA molecule. Okay, so here's our DNA molecule, and I want you to identify the different features of this diagram. So what is this part pointing to? What is this part pointing to? And what is the whole thing pointing to? So remember, DNA is composed of three main ingredients. The first is our sugar and our phosphate, and that composes this backbone that we see here. Sugar phosphate backbone creates this part that holds the whole DNA molecule together. Then, in between each of the two strands of a DNA molecule, you have our nitrogenous bases. So remember, those are the A, T, C, and G, which are connecting each of these strands together using hydrogen bonding. So each of these two strands right here is connected in the center through a hydrogen bond. And you can see the strands because the sugar phosphate backbone is colored differently in this photo. So here, this blue strand is one of the strands, and then this orange strand is the second one, and they're twisted together on top of each other like a twisted ladder. And that's why we call it a double helix, because there are two strands and they're double. It's double strands, right? And so now you have two strands twisting together to make that double helix structure. So before a cell divides, it has to duplicate its DNA in a process called replication. So during replication, here is our original DNA molecule up here. The two strands of each DNA molecule separate. So you can see right here, here's one strand of the DNA molecule, and here's another strand. And before it replicates, the two strands separate. Now, new complementary strands are then synthesized. So synthesize means to create or make. So these new complementary strands are synthesized following the rules of base pairing. So as you see here, okay, here's the original blue strand, and now there's a totally new strand that's being synthesized or made that's based on this original strand. So each strand of the double helix of DNA will serve as a template or a model for the new strand. So this blue strand is modeling what the new orange strand is going to look like. And each DNA molecule resulting from replication has one of the two original strands and one new strand. So when this DNA is replicating, here's the original strand, and then what will happen is you'll get two totally new DNA molecules, but they still look exactly the same as our original molecule. So here's going to be one of the two new DNA molecules, and this will be the second new DNA molecule. So here is our replication fork. Here is where the DNA is going to start replicating. And what will happen is nucleotides, remember nucleotides are pieces, they're monomers of the entire nucleic acid, right? They're going to be added onto the original strand, right? And so these new nucleotides right here are being added on to our original strand, and they're going to make up our complementary strand here in orange, okay? And they're being added on in this direction, right? So here's where the replication fork opened, and then all of these new nucleotides were added on, and they'll continue on down here. Now on this side, the replication is actually happening in the opposite direction. So the replication started down here at this, what we call a replication fork, just like a fork in the road. And nucleotides were added in this direction all the way up. And as you can see here, they're still being added on, and they'll continue and continue on all the way up. So the replication is happening in this direction. Now remember, in order for a DNA molecule to become replicated, the new nucleotides have to match up with the original bases on the original DNA strand. So right here you see this, let's go back. You can see this yellow base is being added. That's a thymine. And remember, thymine always bonds to adenine or adenine, right? So this green one, those are being connected. Now let's see what's on the original strand right here. We have purple, which is representing our cytosine. And do you remember what C bonds to in DNA bonding rules? Well, you might remember that C bonds with G. So as you can see here, this red color is coming in to bond with the cytosine. The guanine and the cytosine are bonding together. And the same thing happens here. So where do these nucleotides come from? Well, there are actually places in your in your cell where there are nucleotides just hanging around on the side. And when it's time for the DNA to replicate itself, those nucleotides will come on in, and with the help of some of these enzymes, DNA polymerase, those nucleotides are going to be added on to create our new orange complementary strand of DNA. Now, let's practice this. So here is our the nucleotide bases of our original strand of DNA, and let's try to complete our complementary strand. Remember, complementary means the matching strand that will be created. So if one of the original strands has the sequence TACGTT, what nitrogenous bases would the new complementary strand have? So let's practice it here. So we know that in our original strand, there was a T on our DNA. And so some new nucleotides are going to come in. And what do we know? What are the base pairing rules for what matches up with T? Well, you might remember that T always matches with an A. What about A? What does A always match with? Well, we know A matches with a T. A C always matches with a G. A G always matches with a C. A T always matches with an A, just like we said here. And a T, again, always matches with an A. So just to remind you, A's always pair up with T's and vice versa. C's always pair up with G's, right? And so whenever the new nucleotides are being added on, they follow the same exact base pairing rules.